Melissa Russell, and I'm a graduate student here at the Institute for Nanobiotechnology at Johns Hopkins University. I'm here today to talk to you about a topic many researchers here at the INBT are investigating: drug delivery. Most commercially available drugs have to pass through the digestive system, from your mouth to your stomach to your small intestine, where they are absorbed into the bloodstream. Once the drugs get to the bloodstream, they have to pass through multiple other obstacles and barriers before they can reach their target. But how can we test if a drug can get to where it needs to go in the body? Ultimately, what makes a good drug? And what delivery challenges do researchers face? We spoke with several faculty and graduate students to find out more.
Many drugs are delivered systemically. That means they're injected into the vascular system and hence are transported to the particular site for treatment. But in the vascular system, drugs can interact with the components of blood. They can be absorbed into vessels or capillary beds or other tissues, resulting in much lower concentrations at the desired site.
So depending on what disease you're trying to target, you might only want the drug to go to one area, and this is especially important in like tumor biology, right? You want cytotoxic agents to only go to the tumor. Obviously, we can't test directly new compounds on humans. That would be problematic. Although, in the past, many drugs were discovered that way. You want your drug screening process to begin with an assay, some test against your target that's very high throughput, that you can test a lot of things, and the readout is very reactive and also very inexpensive.
So in vitro models can be really useful in trying to evaluate how efficiently one can deliver a drug by assessing some of the particular barriers that that drug has to undergo during the delivery process.
An in vitro model gives us an inexpensive and simplified version of a system. In a transfer model, cells are grown on a porous membrane. The cells on the porous membrane and the walls of the well separate the top chamber from the bottom chamber. The drug of interest is added to the top chamber, and the amount of drug in the bottom chamber is measured over time. By using these measurements, we can calculate the rate at which a drug passes a monolayer of cells.
So in this context, an in vitro model, or an engineered model that takes into account part of the system or part of the process, can be really useful in trying to understand some of the details of the delivery process.
The human body, or any animal, actually is a very complicated, interconnected system, lots of feedback loops and things.
We don't know about the physiology of the system, so there is basic knowledge missing that we need.
Current in vitro models lack a lot of the physiological components that are present in the human body: physical geometry, local microenvironment, and blood flow that provides shear stress on blood vessels and capillaries.
In general, it's very difficult to build good in vitro models. It sounds like it should be relatively straightforward to be able to engineer different kinds of platforms to assess the efficiency of drug delivery. The reality is that the platforms we have are quite simplistic and very often don't allow us to address the important questions we want to answer.
One of the things we're working on developing in the lab is a three-dimensional in vitro model, or vascular unit. A 3D vessel is embedded in a collagen matrix and serves as a platform for testing potential drug therapies. One of the real advantages to this platform is that vessels can be grown on a chip about the size of a quarter, and they're optically transparent, which means that we can begin screening drugs directly on the microscope. This allows us to get a much better picture of what's actually happening in a target tissue after a drug is introduced into the bloodstream.
It's very useful to be able to have an artificial vessel, a platform that mimics that part of the delivery process.
So people say if there is a will, there is a way. So we can only hope that a lot of dedication and creativity will pay off in the future.